Great. Thank you to those who are joining us in person and then on Zoom. Welcome to Cardiology Grand Rounds. It's really a true pleasure to have Dr. Cohen join us today. Dr. Cohen did his cardiology fellowship at Brigham and Women's Hospital across the street, followed by a postdoctoral fellowship at Dr. Bruce Spiegelman's lab at Dana-Farber. Since then, he's developed a passion for metabolism, obesity, and specifically built a very robust research program focused on adipose tissue and adipose biology. Yesterday, I named a number of different accomplishments of Dr. Cohen's. He's a wonderful mentor and has trained over 25 MD PhDs and postdoctoral fellows. He really spends about 90% of his time in the lab doing discovery work on adipose biology. The remainder of his time is spent seeing patients in Memorial Sloan Kettering. And his work has really brought innovation to the field of adipose biology by integrating chemistry, hydropodomics techniques, as well as in vitro and in vivo modeling. So very much looking forward to hearing about Dr. Cohen's work.
Hey, I hope everyone can hear me. Thank you all for joining in person. Thanks, RT, for the nice introduction. And thanks for those of you on Zoom. It's been really a wonderful visit so far, and I look forward to meeting those of you who I haven't met already. So today I'm going to give a more translational-based presentation about the role of brown fat in humans. But before I do that, I just want to give you a little bit of background. And please feel free, I'd like this to be interactive, so if people have questions, just interrupt and fire away. Okay, these are my disclosures. They're not in any way related to content being presented here. So the backdrop of our research question, let's see if I can.
I'm floating, yeah.
The backdrop of my research is this question of obesity. So it's been known for a long time that the prevalence of obesity is rising in the United States, but this is not just a problem of the United States or even of industrialized nations. It's really a global problem. There are now more than 650 million people worldwide who are obese, and by 2030, it's estimated that 50% of adults in the United States will be obese, so it will become the predominant phenotype. And as you all probably know, obesity obesity is concerning because it's a major risk factor for some of the leading causes of morbidity and mortality, including cardiac risk factors, coronary heart disease, congestive heart failure, a variety of different kinds of cancer. And then over the past few years, we've learned, unfortunately, about the close link between obesity and severe illness and death from COVID-19. So since this is a clinical conference, I just wanted to give you a flavor of the kind of patients that I see in my clinic at Sloan Kettering. And I think it also highlights how a lot of these comorbidities travel together. So these are two patients I've seen. They're I suppose somewhat extreme examples, but not so extreme that I don't see people like this quite routinely. So the top is a 75-year-old man with obesity, hypertension, hyperlipidemia, sleep apnea, coronary disease, AFib, and congestive heart failure with newly diagnosed rectal cancer. And the reason for the consult was to assess whether he was a suitable candidate for surgery or whether his comorbidities and the risk prohibitively high. The second is a woman who's a 52-year-old survivor of Hodgkin lymphoma, which probably played a large role in some of her downstream problems, along with obesity, hypertension, lipidemia, atrial fibrillation, coronary disease, congestive heart failure, subsequent breast cancer, and newly diagnosed lung cancer. And I show you here both of their body weights and BMI, not to imply that that caused all of their health problems, but it has certainly contributed to their severity. So the question that's motivated us is to understand the mechanisms linking obesity to those comorbid diseases, which is actually not very well understood at a basic cellular and molecular level. So to address that, we focus on the biology of fat or adipose tissue. These are electron micrographs of white and brown fat. White fat is specialized for storing excess energy in the form of triglyceride, which can be mobilized by lipolysis when nutrients are scarce. And one half of my lab is focused on understanding the role that white fat plays in contributing to obesity-associated diseases. But the topic for today will be brown fat, which is this tissue that was neglected in humans until relatively recently, and asking this question of whether brown fat may in fact be a potential answer to protect from obesity-associated diseases, and if so, how this might work. So this is just a cartoon highlighting some of the key features of the cell biology of white and brown fat cells. So as I mentioned, white fat cells are specialized to store lipid. They have this large unilocular lipid droplet. They have very few mitochondria, and they really are there to store excess energy. Brown fat cells actually have a very different cell biology. So they have these small multilocular lipid droplets to facilitate access and utilization of lipids. They have a very high number of mitochondria. Along with the heart, they're among the most mitochondria-dense tissues in the body. And it's the iron in mitochondria that literally gives brown fat a brown color. And these cells and the tissues they contain, rather than storing energy, they can convert chemical energy into heat by a process called thermogenesis. And this is just a depiction of how this works. Some of you may be having unpleasant flashbacks to your biochemistry class. So this is a depiction of the electron transport chain and oxidative phosphorylation. And normally this process generates reducing equivalents, which are passed along the electron transport chain, which results in the generation of a proton gradient, which powers the synthesis of ATP through the complex 5-ATP synthase. What is unique about brown fat is that it expresses this protein called UCP1, or uncoupling protein 1. It's not expressed in any other cell type in the body. And this protein allows protons to leak back across the inner mitochondrial membrane, dissipating that gradient. And the result of that is the creation of a feudal cycle, so that all of the upstream steps are upregulated to try and generate the gradient, which can never happen. Many of those reactions are exothermic, and the end result is the utilization of chemical energy and the generation of heat. So it's been shown in work dating back many years, actually done at this hospital by Brad Lowell and Jeff Flyer, that if you ablate UCP1 positive cells in mice, that results in cold intolerance, not surprisingly, because this tissue is important for generating heat and cold, but it also results in obesity in mice. And it's been shown in a variety of subsequent studies that this process, at least in mice, activating brown fat is associated with benefits, at least in part because brown fat metabolizes a lot of fuels that in excess can be toxic in the body, including things like glucose-free fatty acids and branched-chain amino acids. So for a long time, the canonical role of brown fat is that it would protect newborns from hypothermia. So I'm showing you here some newborn mice that are imaged with a thermal camera that detects heat generation, and you can see the newborn pups are all laying in a pile on top of one another, but this brave young mouse has gone for a little walk, and you can see this very strong heat-emitting signal on its back in an interscapular location, and that corresponds to this heat-generating brown fat. And it's also been known for a long time that newborn humans have brown fat mainly located along the neck and cervical spine, which also serves this important heat generating role. But it was thought that this tissue, especially in humans, atrophied by adulthood and served no real role. In mice, that concept began to change with a variety of studies. I'm showing you one dramatic depiction from a review article. So this is a dissection of a mouse where only the fat is shown and the rest of the mouse has been removed. And this is a mouse housed at 28 degrees Celsius, which is the thermoneutral point for a mouse. And this is a mouse housed at 6 degrees. So mice will live quite comfortably indefinitely in 6 degrees and even colder. And one of the reasons they're able to do this is because they upregulate their brown fat and they brown their white fat so that their fat now goes from an energy storing organ to a heat generating organ. And you can see that in the clear difference in the color of the tissues. We've also learned that there are multiple kinds of brown adipocytes. So in work largely done in Bruce Spiegelman's lab, where I did my postdoctoral training, but a variety of other labs, it became clear that there were two kinds of brown fat cells in mice and in humans. There are developmentally preformed brown adipocytes, which I'm showing you here, that are present at birth and there to generate heat. But there are also an inducible kind of brown fat cell that has been termed beige fat cell. And these cells, rather than storing energy, are able to dissipate chemical energy as well. But the main distinction with classical brown fat is that their activity is highly inducible. So in the basal state, they may look and function like a white adipocyte, but in response to cold, beta-adrenergic agonism, aerobic exercise, or thiazolidinediones, they can turn on this thermogenic or heat generating program. And these cells are present in adult humans. At a molecular level, this is about as molecular as I'll go in the talk. This is controlled by a transcription factor called PRDM16. And in work that dates back now more than 10 years, it was shown that this is the master regulatory factor for brown fat. So if you ectopically express PRDM16 in a white fat cell, that's sufficient to drive the entire molecular and physiological program of brown adipocyte. And PRDM16 actually works as a co-regulatory protein, so it doesn't bind DNA itself, but it binds and bridges other DNA binding transcription factors. And it can actually have two roles. It can activate the expression of brown and beige fat selective genes, but through other protein contacts, including chromatin modifying enzymes, it can also suppress the expression of white fat selective genes. So as a postdoc, one of the things I did was to generate a mouse model that has a selective deletion of this inducible beige fat by deleting PRDM16 only in postnatal adipocytes. What we saw was that these mice lacked beige fat. But interestingly, when that happened, they also accumulated more visceral fat, which is a pathogenic kind of white fat. And these animals had many features of metabolic syndrome in humans, including mild obesity, insulin resistance, fatty liver, and increased adipose and whole body inflammation. And so this work, as well as a variety of other studies from people in this community and elsewhere, really led to this notion of, could thermogenic fat be targeted for therapeutic? And I'd say until relatively recently, the thought was that this would be a waste of time. And the reason was because the prevailing view was that brown and beige fat were important in small mammals and newborn humans, where hypothermia is a major threat to life, but that by adulthood, this tissue was no longer relevant. And that notion began to change, which I'll show you in the next slide. But the first inkling that that might not be the case came from this paper, which was a paper in the radiology literature that I don't think many people read or paid attention to, but radiologists who are reading FTG PET-CT scans, mainly done to diagnose or track the progression of cancer, were noticing that when they did these scans, they would often see very high glucose uptake in these symmetrical tissues along the neck, and that these tissues on CT scan had the imaging density characteristics. And so they referred to this tissue as USA fat or upper supraclavicular fat, and it had this name, but what its role, if any, was not really known. However, several years later, there were 3 landmark papers published in the New England Journal of Medicine, including one from Beth Israel by Aaron Sipis, Ron Kahn, and others, where they showed for the first time that adult humans do in fact have brown fat, be cold inducible, and it has metabolic functions. So this is a figure from one of those studies where they recruited young healthy subjects and did PET scans either at thermoneutrality or after just 2 hours of moderate cold exposure, roughly on the order of about 60 to 65 degrees Fahrenheit, so not freezing cold. You can see this dramatic change where now there's very clear and strong glucose uptake in tissues along the neck and cervical spine. In one of the studies, subjects actually were brave enough to allow people to biopsy these tissues. And on biopsy, these tissues have the cellular and molecular characteristics of brown fat and rodents. And so I'm not going to show you all of the subsequent work. I'll show you some of the key findings, but in aggregate, the work over the 10 years that followed really took 2 forms. One was retrospective studies where people would look at series of PET scans, typically a few hundred scans, and then correlate those, the brown fat status in the scans with clinical variables. And then the other were small prospective studies, generally of young healthy subjects, almost always male, almost always white, in which people were recruited had baseline PET scans, and then were exposed to cold or some other stimulus to activate brown fat, and then some physiological readout.
Hey, Paul, what's going on in the hearts there too?
Well, I think you can clearly see there's increased glucose uptake. Why is increased so dramatically by cold exposure? I don't know. Presumably the heart is demanding more energy.
That's
Yeah. So
pretty striking.
yeah, it is. The other thing you'll notice, which I'll come to, is that almost all of the brown fat is above the diaphragm. So this just shows you some of the knowledge that was gained in these studies. So first of all, in terms of determinants of brown fat in humans, brown fat correlates nicely with outdoor temperature at the time of the scan. So scans done in cold temperature months of the year have a higher prevalence of brown fat than scans done in warmer months. Brown fat prevalence also goes down with increasing age, unfortunately for all of us. Goes down with increasing body mass index, and it goes down with increasing glucose levels. Remember, these are associations, not necessarily causations. Brown fat is also involved in energy metabolism. So in this study, they actually did whole body energy expenditure studies in humans, and they saw a pretty nice association between brown fat activity and energy expenditure. This just highlights the different depots of brown fat in humans. This is work from Aaron Cyphus, who did this initial pivotal work here at Beth Israel, and he now leads a group at the NIH focused on brown fat in humans. And there are 6 main depots of brown fat in humans, all located above the diaphragm. I'll also just mention for this audience, this is not in this slide, and this can be seen on some PET scans, there is a pretty prominent signal of brown fat around the aorta in the thoracic region. So interestingly, in both humans and mice, the thin layer of fat, sometimes not so thin, that aligns that's around the outside of the aorta is very brown in character in the thoracic portion of the aorta. And then as one moves down into the abdominal aorta, it becomes progressively white in character. And what that means physiologically or biologically, I don't think is quite clear. The typical way brown fat is detected, as I mentioned, is with FDG PET-CT, but there have been a variety of other modalities, mainly used in the research setting, including MRI, PET, MR spectroscopy, ultrasound to look for increased blood flow, infrared imaging, which I showed you in mice, and then I guess the cheapest way is to actually measure local skin temperature increased over the neck when brown fat is activated. These are a couple of papers that I think are informative, highlighting the metabolic effects of brown fat. So here is a study in Japan where they took young, healthy males and did a baseline scan to see if they did in fact have cold activated brown fat. So they put them at 19 degrees Celsius for 2 hours, fairly mild to moderate cold, and 27 out of 51 subjects had brown fat. This is a subject with brown fat. This is one without. They also measured energy expenditure, and the people with brown fat had a clear increase in energy expenditure at the colder temperature, which was blunted in those without brown fat. They then took the subset of people who had brown fat, and they did this regimen where for 6 weeks, they went to 17 degrees Celsius for 2 hours a day, and then they looked at the effects. And what they saw was that there was a statistically significant decrease in fat mass at the end of the 6-week study, and that correlated with an increase in induced thermogenesis. And then finally, this is one of the few studies that actually has looked at brown fat in people who are not so-called normal. So here is a study from the Netherlands where they recruited 8 subjects with type 2 diabetes, and they had them exposed to cold for 2 hours a day at 14 to 15 degrees Celsius, so a little bit colder 10 days. And then they did hyperinsulinemic euglycemic cramp studies. And the main take-home point, as you can see here, the glucose diffusion rate rises afterwards, consistent with improved whole body insulin sensitivity. And in this panel, what they're measuring is whole body glucose disposal, which also increases after the 10 days of cold exposure. So I think all of these studies clearly suggest that brown fat plays a role in glucose metabolism and insulin sensitization. But when Tobias Gescher, who is an MD from Germany who joined my lab. He was really interested in asking more broadly, does brown fat protect against this constellation of comorbidities associated with obesity that we see in humans? And he recognized that one of the limitations of the existing body of knowledge is that the size of the studies was not really large enough or powered well enough to ask this in a comprehensive manner. And so to address this, we took advantage of the natural variation in human brown fat that you can see in clinical PET scans. Unlike the interventional studies I showed you where people are cold exposed to try and activate brown fat, in clinical scans, the opposite is done. So subjects are warned, they're told to refrain from exercise and caffeine. The goal is to suppress endogenous brown fat activity because, of course, if you want to image cancer, you don't want some background signal, especially if cancers in the same region. Even with that, you can see, though, in these clinical scans, some people have quite a lot of brown fat and some people have hardly any. So we took advantage of the fact that at Sloan Kettering, which is across the road and where I see patients, they do probably more than 10,000 PET scans a year at their various sites. And because they were the group that initially described this USA fat, they've had a long-standing interest. And so when they read their reports, they always comment on whether they see brown fat. And so what that allowed us to do is to download the reports rather than manually rereading the scans, which would have never been possible. And based on a search for the term brown fat, to categorize people as either brown fat positive or brown fat negative. And then we could link patients' brown fat status to all of the information in their electronic health records. And so that allowed us to review over 130,000 scans for more than 52,000 patients. So by more than probably 2 orders of magnitude, the largest study we were aware of at the time. Now, I mentioned earlier that brown fat is associated with age, sex, BMI, and outdoor temperature. And so to control for those covariates, we used propensity score matching, in which we matched people with brown fat, those without brown fat, based on these 4 variables. And when we did that, we ended up with a cohort of about 5,000 people with brown fat and roughly 10,000 people without brown fat. So because they're matched by BMI, that controls for body weight and obesity, which becomes relevant. So when we then linked that to their diagnoses and their health record, we saw that people with brown fat had a really dramatic reduction in the odds of a variety of prevalent and serious cardiometabolic diseases, with the most dramatic reduction in type 2 diabetes, consistent with what had been learned about glucose and insulin sensitivity. But what had not been described before is this pretty prominent reduction in the odds of coronary artery disease, congestive heart failure, hypertension, and cerebrovascular disease. And because this is matched by BMI, these effects are obesity independent. We also did an analysis where we grouped people by BMI into normal, overweight, and obese. And if you focus on type 2 diabetes here, you can see people with brown fat are in brown and those without are in blue. And you can see in each weight category, those with brown fat are significantly protected from type 2 diabetes. So brown fat doesn't only protect people who are normal BMI, but even in the obese range, people with brown fat have a similar prevalence of type 2 diabetes as someone who is overweight or normal weight without brown fat. Yes?
I'm just curious because you know, very rough estimate of obesity adenomas. What is the association of brown fat and visceral adenomas? Are they sort of
You gave me a great segue to something I'm going to show you in a couple of slides. So if it's okay, I'll answer your question in a few slides. But if it's not answered, please stop me again. So we also looked at laboratory values in these patients, and we saw that fasting glucose was significantly lower in people with brown fat. We also plotted fasting glucose as a function of BMI, and not surprisingly, in people without brown fat, there's a clear relationship where as BMI goes up, fasting glucose values go up. We saw the same thing with triglycerides, but in people with brown fat, this relationship is very much flattened. So we have since really been focusing our efforts on understanding what's responsible for these benefits associated with brown fat. So we asked a variety of questions. The first, which I think gets to the question you asked, is brown fat associated with a healthier distribution of white fat and decreased ectopic lipid? Second are the pleiotropic effects of brown fat mediated by the secretion of humoral mediators. And then finally, how is brown fat regulated, and are there genetic determinants of brown fat quantity and activity? So to address the first question, what we did is we went back to this cohort, and in a subset of patients, we manually reviewed the scans, or I should say Tobias did this. It took him about 6 months of sitting in a reading room and doing this. And we took advantage of the fact that on the CT scan, if you look at the L3, L4 region, there are established methods where you can use to actually quantify the amount of subcutaneous and visceral fat. And so we looked for the relationship between brown fat as a binary variable as well as a quantitative variable and subcutaneous and visceral fat stores. And this is the main finding here. So what we saw on the y-axis is fat area, and on the x-axis is BMI. You can see that with increasing BMI. In blue are people without brown fat. Both the subcutaneous and visceral fat area go up. But in people with brown fat, they tend at each matched BMI, they have relatively more subcutaneous fat, which is the healthier kind of fat, and less visceral fat. And that's also seen here in this regression analysis, where we see that as brown activity goes up, the amount of visceral fat goes down, and this is controlling for a variety of other variables. So we think that at least one reason for these benefits that are associated with brown fat is that it's associated with a healthier distribution of brown fat at the same BMI. However, take this for what it's worth. We did also in these regression analyses look to see how much of the overall effect of brown fat could be accounted for by this shift in body fat distribution. And statistically, this could account for some, but not all of the effects of brown fat. So we believe this is part of the story, but not the whole story. We also, in this same study, took advantage of the fact that on the CT scans, there are methods to look at liver density, which correlate with liver fat, so that a higher liver density is correlated with lower hepatic lipid. And by doing that, we saw that people with brown fat have significantly higher liver density, meaning associated with less lipid storage in the liver. We also recently published this paper looking at cancer outcomes, and the reason we did this study is because in mouse models, there have been a variety of studies, and as a result, there's this prevailing notion in humans that in mice models of cancer, activating brown fat is associated with a worsening cachexia syndrome and worse outcomes. And so because of that, many people felt, okay, well, brown fat may be associated with all of these benefits, but in the setting of cancer, maybe it's actually a bad thing. In our study, we did not find that to be the case. So we looked across all cancers as well as the most prevalent cancers in which we had PET scan data, and there was no association between brown fat and increased cachexia. If anything, brown fat was associated with less cachexia. And then we also looked at survival, and this was shy of statistical significance, but across all cancers, there was a trend towards brown fat being protective value, I think. So let me now turn to the other mechanism, which I touched on in the research talk yesterday, but I'll give you a bit of a different perspective now. So if brown fat's benefits can't entirely be explained by a healthier white fat distribution, how else might it work? And so we've become really interested in the thought that perhaps the benefits of brown fat in some or maybe in large part might be mediated by humoral mediators. So what might those be? So this work has been done by a variety of really talented people in my lab, initiated by Chan He, who's a graduating MD PhD student. He's also destined for the world of cardiology. Will, who's a PhD student now at MIT. Kaya, who's a postdoc. Corey, who's a research assistant. And Samir, who has been in my lab as a Sarnoff fellow and will be joining you at BI as a medicine resident. So we've had a great team to address this question, but before I talk about the data, I just want to show you at least one piece of evidence that I think at least suggests this is plausible. So for those of you who were there yesterday, I apologize, you've seen this already, but this is a study in mice where they used a mouse model of type 1 diabetes. So mice were treated with streptozocin to ablate pancreatic islets. That leads to very significant hyperglycemia with blood glucose levels above 400. They then took a very small amount of brown fat from young animals, roughly 50 milligrams of tissue, transplanted it into the animals, and that resulted in durable, near complete correction of the blood glucose values. There was no loss in body weight, so the animals weren't sick or starving, and the animals continued to completely lack insulin. So somehow brown fat can largely correct type 1 diabetes in an insulin-independent manner. What that is, I think, begs an answer, but it seems very unlikely that this could just be explained by brown fat taking up so much glucose, this 50 milligrams of tissue, to completely normalize blood glucose levels. So other reasons for looking at this, we know by bioinformatics searches that fat cells are predicted to secrete more than 1,000 unique polypeptides and probably an even greater number of small molecule metabolites and peptides. And although there are some very well-known molecules like leptin and pectin, adenosine, and others, the vast majority of these molecules, as well as their targets and functions, have not been characterized. One of the main limitations is technology. And we think that if we can assemble such a list and figure out what these proteins and molecules are doing, we may be able to explain how brown fat mediates its benefits and perhaps even identify new therapeutic targets. So this highlights one of the challenges at a proteomic level in a typical analysis. So proteins in the blood exist over more than 10 orders of magnitude in concentration. And so if one just wants to do standard mass spectrometry from serum or plasma, one will detect a lot of albumin and globulins, but it's very difficult to identify hormones. We've been using a variety of chemical techniques, cellular models and mouse models. I'm not going to go into this in great detail, only to mention it's a metabolic labeling technique where we can introduce unnatural amino acids with chemical handles on them that allow us then to take media from cells or blood from animals, use those handles to enrich proteins in a self-type selective manner, and then identify them by mass spec. So we published this recent paper in eLife, which highlights our initial results. We identified more than 600 unique proteins, about 350 of which show enrichment in the secretion of different kinds of adipocytes. And when we did functional analysis, we saw that those secreted proteins were involved in rather different functions. The proteins secreted by visceral white fat cells are associated with activation of the immune system, perhaps consistent with the pro-inflammatory role of visceral fat, whereas proteins secreted by brown fat seem to have a prominent role in neurogenesis guidance, which is not surprising. But we have also been really moving away from cells to in vivo studies, mouse models, using some of these same chemical labeling approaches, as well as bulk approaches in mouse models. And then what I want to focus on today are our translational studies in humans. So I want to take you through a couple of the studies we've done. So the first is something we refer to as the cold vest study. And so what we did is we recruited 20 men and women, less 28 years of age or younger. We specifically picked these demographics because these are ages and BMI when the prevalence of brown fat is very high. So we wanted people who were very likely to have cold activated brown fat. We did not, we excluded people with any comorbidities, any medications, any illicit drug use in a recent period. And then we used a customized cooling protocol that's been described by Aaron Sipes and others, where we used a cooling vest. It looks a little bit like a life vest that you plug into the wall. It has cold water circulating through it, and you can adjust the temperature and make it successively colder. And when the subject starts to shiver, we then raise the temperature by 2 degrees so that they're not shivering during the study. We don't want to be measuring muscle shivering. And then they sit at this temperature for 3 hours. We measured body composition as well as some clinical labs, and then we got research blood samples at baseline and after 3 hours of cooling. And in collaboration with Robert and people from his group, we have done some really deep plasma profiling, including looking at the proteome by SomaScan and Olink, as well as looking at metabolites and lipids in an unbiased manner. So this has been a fantastic collaboration. And the goal has been to identify factors that are commonly regulated by the acute activation of brown fat and then to intersect that data with data from our transcriptomics or our mouse data to create a list of real prominent candidate batokines or bat-derived molecules and to begin to functionally study them. So I'm just highlighting some of the key findings so far. We saw quite a lot of molecules that are changing significantly between baseline and after 3 hours of cold exposure, proteins, metabolites, and lipids. We're now doing some control studies to control for the effects of fasting, and I'm happy to address that if people have questions. And we're also obtaining blood samples from a validation study from a separate cohort where subjects have had baseline and post-cold exposure PET scans so that we can actually confirm that the trend in these metabolites or proteins correlates with brown fat activity and maybe even look at the quantitative relationship. So hopefully more on that to come. I did want to highlight a couple of pieces of very early results. So this is a volcano plot of metabolomics. You see quite a large number of metabolites are upregulated in blue or downregulated in red. And I'm just showing you a couple that are kind of encouraging as sanity checks. So one is this lipid called dihome, which was identified and published by Yuat Sang at Joslin here. And they identified it as a cold-induced batokine in mice and humans that regulates metabolism and brown fat locally, but also acts systemically to regulate metabolism in a variety of other tissues. So matching her reported findings, we also see dihome increase on cold exposure. And then the second is this metabolite that's been studied by Robert's group called DMGB. Levels of this metabolite are positively associated with incident diabetes and liver fat. And so it was nice to see that levels of this so-called marker of unhealthy metabolic state decrease on cooling. Whether that these metabolites, this data on its own does not indicate these metabolites necessarily come from brown fat, but they're at least associated with the situation that results in activation of brown fat. We also recently kicked off a related study, which is our best attempt to study chronic cold exposure. So this study did not come up by design. We were actually contacted by people in the Minnesota area who were aware of the research interest in brown fat, and they're very passionate ice water swimmers. So this is a real picture. They make holes in a lake in the Minneapolis area, and they immerse themselves in the water. They really enjoy it. When you pay them to do this, they feel it provides tremendous health benefits. And speaking to them, actually, the most prominent benefit that they report is mental health benefits. So, the thought of doing this gives me great anxiety, but the common thing that they say is that it actually has a managing anxiety effect on the days when they immerse. They just feel very calm and at peace with whatever the world is throwing at them. So they actually number a few hundred individuals, and they're really well organized. They collect data on the temperature of the water, the temperature of the area. And because it's a public lake, for health reasons, they have to scan a barcode on their phone each time they go to swim. So we have pretty rigorous data about the amount of cold they're exposing themselves to. And so the way the study has been designed, and this is in collaboration with Betsy and we hope with Robert's group again, we are collecting samples and phenotyping these people in the winter when they're doing cold immersion, and then again in the summer when they're not. We also are collecting samples from controls who are friends and family members who live in a similar environment but do not do this. And in addition to doing the same plasma profiling on a subset of individuals, we'll do deeper metabolic profiling, including PET scanning, hyperinsulinemic euglycemic clamps, and taking fat biopsies for molecular analyses. We've already collected samples from about 20 patients, and we're hoping to collect the rest of the winter set before it warms up in Minneapolis. So in the last few minutes, I just want to turn to the last area, which is really one of our main interests going forward, which is to understand how brown fat is regulated in humans, and are there genetic determinants of brown fat quantity and activity. So the idea for this really comes from looking at these scans, which I showed you before, and you can see this pretty dramatic variation in brown fat across individuals, even people of similar age, sex, BMI, and other characteristics. So there were some suggestions in the literature that it wouldn't be crazy to look at the genetics of brown fat. So if you look, this is data from the CITES paper, which is a scatter plot of brown fat mass in men and women. As I mentioned, women have more brown fat than men, but there are a couple of individuals who have exceptionally high amounts of brown fat. And in other studies, looking at these so-called exceptional variants is often a way to identify rare genetic variants or mutations that are associated with prominent phenotype in an Mendelian fashion. The other bit of data comes from a study done in the Netherlands where they looked at brown fat volume in about 100 people living in the Netherlands who are either of Caucasian or South Asian ancestry, and they saw matched for other characteristics that people of South Asian ancestry had significantly lower brown fat volume. Feel free to take a picture if you wish. Yeah, so you know, this is the only such study I'm aware of, but one of the things that was mentioned, which I guess is an interesting notion, this is a population which has an increased risk of metabolic syndrome and diabetes, but not necessarily associated with the type of obesity we see in Americans. And so perhaps the difference in brown fat would be somehow related. At any rate, we've been taking a variety of approaches, which I just want to highlight. One is that we've gone back to our Sloan Kettering cohort and looked at individuals. This is a scatter plot of brown fat activity by age category, and in red are people with brown fat activity more than 2 standard deviations greater than the average for their age, again with some of these exceptional variants. And a subset of these people have had their exome sequenced as parts of other studies, and so we've been able to obtain that data. And so we're looking for rare variants in people with exceptionally high amounts of brown fat activity relative to controls to see if we can identify any candidates that we can then test. And then the other approach we've been taking is to look at well-defined cohorts with extreme phenotypes. So the Turkish obesity study is a study in Turkey in largely consanguineous pedigrees where people had BMIs greater than 40. And they had their exome sequenced. This includes about 1,000 people. And so the hypothesis here is that perhaps there are mutations in brown fat genes that over time lead to decreased energy expenditure and a tendency towards weight gain. And then on the converse, we've been collaborating with Dr. Baruchi in the UK, who's led the STILT study, which is a study on the genetics of thinness. So they have sequenced the exomes of nearly 4,000 people who are thin with a BMI below 19, but no underlying health problems. And the hypothesis is that this cohort may be enriched for people with variants that lead to increased brown fat activity and increased energy expenditure. So far we've been taking both a candidate gene approach as well as an unbiased approach. And then we've been doing validations in this BioMe database in collaboration with Econ. And I should say Robert's group is involved in all of these efforts. So the BioMe database is a database at Mount Sinai in New York City, which captures all of the diversity of New York City, and they have exome data on about 35,000 patients linked to everything in their electronic health records. So it's a great way to try to validate our data in these that we find from these cohorts with extreme phenotypes. And then we can do things like PWAS analyses, for example, to see if variants that are seen in both are associated with fashion. So we've identified a number of candidates we're very excited about. And some of them we've gone all the way to mouse models. And so this just shows the pipeline we're following where we pick these candidate variants, study them initially in cellular models to look at their effect on brown fat function. And then with those that have promising readouts, we've made mouse models, either knockout mouse models or knock-ins of these human variants to study the effect on whole body metabolism. So that's all I have for you. Just to summarize, with this backdrop of the medical problems associated with obesity, we think that brown fat is associated with pretty significant protection from cardiovascular and metabolic diseases. And the benefits may be conferred by humoral mediators, and we're eager to identify what those factors are and show how they work. And that secondly, thermogenic fat volume and activity may be under genetic regulation if we can identify the key genes that will provide us with another layer of understanding and perhaps new therapeutic targets to try to reverse this situation. So I thank the people in my lab who led the work. We've had a great group of collaborators, including a number of people who I've enjoyed visiting with the last couple of days. And thank you all for your attention. Happy to get any questions.
Well, thank you, Paul. We do have one question on the Zoom, but we'll start with the top one.
Thanks for a great talk. I mean, one of the biggest rages is cryotherapy, cryo chambers.
Yeah.
I mean, I haven't been asked about it much in clinic, but
Yeah.
is that somewhere that people are going to be reaching out to you, or are you going to be reaching out to them, or?
People have been reaching out already. Whether or not it leads to benefits, I think is unclear. From what I've shown you, it certainly might be.
Yeah.
We obviously need a lot more data. And I think what I stress to them, conservative, is that everything we've seen in the human are associations. They don't necessarily imply causations. And so for us, those are starting points. Now go back to animal models where we can establish causation. But many people, even without a physician's advice, because of these other benefits, are practicing some form of cold exposure. And I suppose in our ancestral life, these periods of cold exposure and warmth were probably more what we were used to, and so perhaps there really are.
Obviously, the same comment, Paul, because I was watching a month ago on National Geographic, the Helmsworth brothers all swimming in, Arctic temperatures in Norway, I think it was. And
Yeah.
I mean, on the one hand, it makes sense. They're also all very healthy and fit appearing. So the people who are going to go to the cryo chamber are the same ones who buy like the thing that vibrates your abdominal walls. But it is, it's everywhere.
Yeah.
It's everywhere.
Yeah, nice to see you, Mike. It is, it's an interesting notion. There was a small study published a couple of years ago by Camilla Sheila, who's a brown fat researcher in Copenhagen, and she did a study of about 8 individuals who swam in the North Sea during the winter. It was a little bit different because they were doing these cycles of swimming and then sauna back and forth.
Yeah.
But they did find on scanning that these people did have increased brown fat activity, and they measured, a variety of blood values. But with 8 people, they were.
Thank you for your talk. Thank you. I'm curious to see, that you showed that there are sex differences in brown adipose tissue on, and I'm interested to see whether you also looked at whether brown fat, the effect of brown fat on metabolic dysfunction is modified by sex. There's some data showing that, for example, women with obesity or with visceral adipose tissue are sort of more susceptible to some of the cardiovascular.
Yeah, we have seen in some analyses that the findings we reported are true in both men and women. But you're right, there are important sex differences, and they're at a more basic level in terms of why there are sex differences. It's been known for a long time that sex hormones, even in basic cellular studies, regulate brown fat activity. Testosterone actually prominently suppresses the gene expression of one another genes in the thermogenic pathway. The other question that comes up a lot is, what do we see in terms of racial and ethnic differences? And how people, unfortunately, it's true, the cohort at Sun Kettering is rich as for us is not rich in diversity. So the great majority of the people in our study were white. So we would love to look at this in other populations, but because most institutions don't make a habit of reporting brown fat status in their reports, it would entail either someone manually reviewing scans, and we've not found anyone willing to do that, or using some sort of computational approach, which I think could be possible, but we just need to find a partner.
Question actually related to that point. So there was a beautiful paper in Nature Medicine, but all the patients have cancer.
Yeah.
And so I was curious as to whether you think you'd have the same PET scan findings on
Yeah.
cancer.
Yeah, so we had the same question and as did reviewers. So I don't have a perfect answer, but a decent number of the patients in that cohort have ostensibly been cured of their cancer. Many of them are with lymphoma who then have annual PET scans as surveillance. And so when we did an analysis on those with inactive cancer, the findings don't true. Something we were really interested in doing, maybe if you know anyone, please let me know, is since PET is used also in cardiac imaging and stress testing, we were wondering, assuming the scans get high enough into the neck, we maybe use those scans to capture brown fat status in a different kind of population and then correlate it with cardiovascular measurements. But again, I have not yet found anyone who has a data bank of scans with brown fat status.
Great talk, Paul. Was it back in the 20s when one of the diet interventions was this, what was it, a coupler, right? A mitochondrial dinitrophenol, right? So is that, so maybe tell that story,
Sure.
and is that a warning though about overactivating this pathway in terms of what could happen for weight loss?
Sure. Yeah, so it's a really interesting story. I don't know if you guys are aware. So in the this first came to light, I think, even before the 1920s. So it was observed that people who are working in munitions factories in World War I, many of them lost a lot of weight that couldn't be explained, and they felt hot all of the time. And they eventually figured out this component in the munitions, dinitrophenol, was responsible. And so largely starting from that, dinitrophenol was used as a weight loss agent. It was approved by the FDA in the 1920s and the 1930s. Thousands of people took it, very effective at losing weight, but it was banned because there were a high rate of complications, including fatalities, presumably completely unchecked, uncoupled respiration in every tissue in the body. So that approach probably is not wise, but about 10 years ago, Jerry Schulman's group at Yale published a paper where they made a version of dinitrophenol that's a sustained release version. So it leads to the same kind of uncoupling, but not at the same level of magnitude. And they did studies in small animals and large animals showing metabolic benefits and safety. And I believe there's a company centered around that now where they're looking for testing. So I think we'll see how.
This is awesome talk. Are there clues about, I mean, the tissue is really, brown fat tissue is really dynamic, which is so profound. So are there clues about how it's sensing glucose and how it's sensing temperature to change, how quickly it's proliferating and stuff?
So great questions. The temperature part has been studied. So the thought is that temperature is sensed peripherally in the skin by the sensory nervous system that leads to a signal to the brain. Specific brain regions have been mapped in mouse models, then send urine output to the sympathetic nervous system, and postganglionic sympathetic neurons densely innervate brown fat, and their nerve projections release catecholamines, which activate brown fat. So that's how the cold temperature is sensed. In terms of glucose, to my knowledge, that hasn't been addressed. I actually have a PhD student in my lab who's interested in that exact question in terms of how brown fat couples nutrient sensing, both its thermogenic and secretory properties.
We have one question, and I'll just read it briefly. It's interesting. So from Warren Manning, fascinating presentation. Thanks for educating us. And there is data on excessive heat, such as prolonged time in a hot tub or sunbathing, that it may be detrimental with regards to brown fat. And can prolonged daily cold exposure be a non-pharmacologic painless weight loss therapy?
Yeah, thank you. Thank you for the question. I'm not aware of any data in humans about prolonged warm temperature suppressing brown fat, but that's certainly what you might expect. In mouse models, though, that is definitely true. So if you house mice at thermoneutrality, it completely suppresses brown fat thermogenesis. And if you dissect these animals, brown fat now has a pale color and it looks by eye, even on gross dissection, much more like white fat. I think many people are interested in whether cold exposure could be a therapeutic modality. Some people have even talked about as a broad public health measure, if we can, start movements to lower the thermostats in office buildings and public housing and so on. That's of course controversial. I know my own marriage has been threatened many times by battling over the thermostat. So I think the problem with that, as appealing as it is just that many people don't like being cold and we like being warm, which is why we wear sweaters and coats and so on and ramp up the thermostat. But perhaps at least being moderately cool part of the time would actually be beneficial.
It's also like climate issues.
Yeah.
Warming is a just public health issue for
All right, thank you so
Thanks,
much.
everyone.